already familiar with, right? So to how to write discussions for the thesis, and finally it's going to lead your conclusions. Tak bergeraklah pula. Okay, let's recap. Where is the discussion in your thesis? So basically, discussion will be number 5 in the thesis. You should have the title, abstract, introduction. Introduction basically 1.0, and then material and methods of the missing literature review. Basically, introduction, material and methods. Before that, you're going to have literature review, material and method, and then you're going to have the result and discussions. Okay. Lepas tu acknowledgements, reference, and appendix. So this is the content of your thesis. So what should be in the result? The result should present your findings. So how to present the findings? You can present the findings in terms of figure, or you can write in the text. Basically, people like to have the pictorial, which is like picture, right? So it's better for you to convert the data, the text data, into the figure or into the tables, into diagrams, so that people can capture very fast. Instead of you write into the text, that will be longer in the text position, right? So better to put in figure or text or table, not the text. Text should be there, but not much, right? So when you use figure or table, it's going to help the reader to understand the data very well, and then they can appreciate your findings. If you don't understand your findings, how come they appreciate your findings? In particular, when your findings answering the question about the hypothesis, right? So that will be very meaningful for the readers, because you're answering the hypothesis. And when the peers read your thesis, they say that this is the one they're looking for. Gitu. Alright, canggih tak? Kalau tidak ada, if you present the data in the wrong form, you might miss that opportunity to be like the impact of your research paper or your findings on the hypothesis. Okay, clear? All right. So basically, we have to choose either to present the data in the table or in the figure. How to choose? Table or figure or text? Based on what? If you're looking for the trend either decreasing or increasing, or you are comparing between two. For example, you're comparing between A and B. So it's better to use figure. Because figure, people can capture the figure faster than table. But if you are detailing something, for example, you are measuring proximate analysis, you are summarized from maybe journals relate to your paper for those who doing the systematic review. So it's better to use table, because if you convert into the figure, you cannot you cannot transfer the knowledge better when you transfer into the figure form. So if you detail out something, better to put in the figure, in the table form, because the table can be more detailed on that one. Okay, right. What should be in the text for the result? So result first, and then followed by discussion. So now we're going to learn how to write the result. So just imagine you have your data in front of you now. Okay, so what you should write in the beginning of the data in the thesis? Firstly, you have to write a this is my style. Many lecturer, they will have their own style. Many students, they have their own style. But the style doesn't, it's not really rigid. But you can follow many style. But because the content, the component of the result and discussion should be there. The first one, you should have a briefly describe of the experiment. Okay, so the description should do without describe detail as in the method. Remember, in methodology, you already described the experiment, right? How many sample you need, how many sample you add with the reagent and so on. So don't repeat again here in the discussion part. In the result part, just tell the brief. For example, if you do antimicrobial, you measure in a microbe. So just mention microbe. Several microbe has been quantified in the food sample. So just brief like that. Don't go detail how many gram of tissue you put in so much. You measure using the selection medium and so on. Just a very brief statement. The first one, and then you report your findings. You report your findings. Basically, your findings something that you already simplify into the figure or in the table form, but you repeat again. You repeat again the important points. So how to select the important point? Basically, we will talk about who is the highest, who is the lowest, who is the trends, what is the range. Okay, so that will be means that they call as maksud tersurat. Maksud tersurat itu macam mana? Maksud tersurat means that if people read the figure or people look at the table, what they understand, what they're looking for. So itu maksud tersurat, tersurat on the table part, on the figure part. Tersirat is something that you have to do comparison. You can, like, it's like you make secondary analysis of the data. So that's tersurat. So the first on the result, you must explain tersurat, part yang tersurat. Okay. So example, this is an example. How you going to explain the result? Maybe you can explain from the highest to the lowest, from the most to the least important. Okay, from the simple to the complex, from the complex to the simple. But the sequence or the trend should be same for all results. You don't change it. For example, 4.1 you are mentioning from top to lowest. 4.2 you mentioning from lowest to top. Jadi pening. Okay, so don't reverse the thing. Make it a trend, a consistent trend. So that's what we call as consistency. Right. Also, you can discuss based on organ to organ, chemical to chemicals. If you're doing for example, if you have different subtopic, so you can use according to subtopic. For example, like Shuhada, she's doing the knowledge, attitude, and practice. So she can break into knowledge one part, attitude one part, practice one part. Okay, so it's depend on subtopic. All right. So how to write the result and discussion. Ini contoh dia ni. There are a few styles to write the result and discussion. This is one of the style that I like to use. So there are many other styles, as I mentioned earlier. There's nothing wrong and right as long as you complete all the elements. Okay, basically this is to write the result. You must have 3 elements. The first element will be talk about the location of your result. For example, this one is the location. Say that a statement that locates the figure of the table. 2. The location of the result. 3. The location of the result. 4. The location of the table. 5. The location of the result. 6. The location of the result. 7. The location of the result. 8. The location of the result. 9. The location of the result. 10. The location of the result. 11. The location of the result. 12. The location of the result. 13. The location of the result. 14. The location of the result. 15. The location of the result. 16. The location of the result. 17. The location of the result. 18. The location of the result. 19. The location of the result. 20. The location of the result. 21. The location of the result. 22. The location of the result. 23. The location of the result. 24. The location of the result. 25. The location of the result. 26. The location of the result. 27. The location of the result. 28. The location of the result. 29. The location of the result. 30. The location of the result. 31. The location of the result. 32. The location of the result. 33. The location of the result. 34. The location of the result. 35. The This is the example. Figure 1 shows the level of caffeine as adenosine inhibitors. Okay, so this is the location, figure 1. And always remember, when you talk about the location, figure 1, table 1 should be in present tense. Okay, remember, should be in present tense. Don't say that figure 1 should. Tu dia macam past tense. Tak boleh. Because your data will be present in the figure 1 forever. Starting from the reader read your data now until forever, is going to present in the figure 1. So you have to put in the present tense. Okay, and then second element should mention about statement that present the most important findings. Basically, we start from the most significant. What is the most significant? Okay, and then you describe the least significant. For example, this one is caffeine was somewhat more potent than theophylline in preventing leaf eating. This is I think this is about the animal, something like that. Okay, so this example, caffeine was, why the authors use was, because this is the findings from the research. So in your study, for example, you have your own findings, so the statement should be in past tense because you already have the result. You conduct the result, and each time you mention your result should be in the past tense. So it means that you already complete the study, and then this is the result from the study, right? So if you mention the result in the present tense, what reader will think? They will think that the thing is still ongoing, right? So you haven't completed your study yet, still ongoing, and then why you have to write the thesis if your research haven't complete yet? Okay, so that's why you have to write in the past time. And the third element, the comment of the result. So you elaborate on the result. For example, in contrast, caffeine has been reported as well to be 10 times higher weaker than theophylline as adenosine antagonist. I then put everything in 3 elements. You can see here. Okay. So if you, when you write the thesis, I suggest you to use different color like this one. Element 1, red color. Element 2, blue color. Element 3, green color. So element 3 basically is talk about the discussion, your findings as compared to others. That's why they have the reference like this. This is a reference. So when you have different color like this one, you can clearly see that your write up for the result is complete. If you have 3 colors like this, minimum is 3 colors, right? The length is depend on the content. It could be longer. It's depend, right? Okay, second style, you can write the result when you combine element 1 and element 2. So this will make your thesis become shorter in terms of text. Why? Because you already shortened the element 1. You don't have element 1 on the sentence. You just use figure 1. For example, like this one. Caffeine was somewhat more potent than theophylline. It's directly explain the result in preventing leaf eating, figure 1. So the reader will say that, okay, I will refer to figure 1 like this one. And then in contrast, caffeine has been reported elsewhere to be 10 times weaker. This is explanation of the result with the reference. Okay, so you explaining your findings should come up with the reference for this one. All right. I see pula. Okay, so I'm giving you dah nak masuk maghrib dah ni kan? Maghrib pukul apa? Pandai sambungan ni, okay. Lepas ni, after we have a maghrib prayer breaks, So please write 3 elements for your thesis. So take one data and then write 3 elements, and then you can share with us in the chat box, or you can share directly personally to me in the chat box. Okay? After this, during the break, not now. So now we're going to see. All right. So this is how you write the table. What is the most important for the table is the legend. The legend should be on the top. That for table. How about figures? The legend should be at the bottom. Okay. So table on the top, figure at the bottom. So must remember with them. Okay. And then when you talk about the figures for the thesis, you should have 3 rules regulation. Why is 3 rules regulation? I'm going to mention. So I will change this one. Okay. Salah table ni is not follow the we call the format of the thesis.
Tapi dia lembab dah.
Siapa tu? Off your mic. Okay, I should follow the 3 rules regulation. Mana ni punya ni? Borders. What is the 3 rules regulations? Who knows? For the figures, for the table, sorry. Let me share the screen, and you can look how I do it. Right? Okay. So what is 3 rules regulation for the table? The first one, you must have the 1 line on the top. And then this is, for example, this component. This is the values in percentage. Okay. Three rules regulation for the table. Kenapa dia ada tak nampak ni? Okay, I have to remove the shading. Hilang tadi punya word dia. The first one, you have the line on the top. So that is the first one. The second rules, line at the bottom. Okay. And the third rules is the line on the heading. Oh, silap. And no borders. Top. Okay. Oh, silap ni. Sini kena ambil line. Borders 2. So you have to play around border top and bottom. Okay. So it should be like that for the table, right? You must have 1 line, 2 line, 3 line. So that's all. So don't put the table with the all line like this. For example, all borders. Lagu ni. So don't put like this, right? So this is not for the thesis. Thesis should be have only 3 line. Hilang dah line saya ni. Line like this. Mana line dia ni? Okay, all right. So let's see how we describe. This is the example. How we describe the result. Cantik. And then the component on the left side should be in the left justify. The right side, the far right should be in the right justify. The one in the middle should be center justify. Okay, for example, I insert one value. Okay, it's like that. And then let's say I put this value for the one in the middle. So this value should be centered like that. Right. And then, okay, when you talk about the table, it should come with the means plus minus standard deviations. So there is the means. Huh? So how get the mean? How to get the mean? When you talk about the value lah, but you talk about the, for example, after this. So the mean come from the average of minimum 3 readings plus minus standard deviation or standard error mean. You can use both, either one. But you should, on the top, you should mention percentage, or at the bottom, you should mention percentage, mean plus minus standard deviation or standard error mean. Okay, and then should follow it by standard deviation or standard error means with how many decimal point. So how to determine what is the good decimal point? Based on what? Can I use this 4 decimal point? Or I can use this 4 decimal point? Based on the sensitivity of the instrument that you use. So that will determine how much the decimal point. If my instrument cannot give me this reading for the ash content, for example, 2.11%. So if my instrument cannot calculate to this 1, so I cannot use this one, right? Only if I just can measure the value 2 only, so means that I have to write 2.0. That's it. Plus minus this one. This standard deviation should follow the numbers of this one, of decimal point based on the value. So if decimal point, if the value cannot measure up to this 4 decimal point, so only 2 decimal point I can measure it. For example, I have this one 0.21. So this decimal point standard deviation will follow the same one with this one. Something like that, right? So there is one rules. I might be wrong here because I think I do some mistake on the measuring the numbers of the standard deviation. Okay. So how to describe this table? The first one, you should locate this table, which is first element. This is how first element. Table 1 shows the carbohydrate content in the sweet potato was so this already second element. In the sweet potato was the highest with 90% carbohydrate, the highest 90%, followed by moisture content 65%, and so on and so on. Right? So I already have first element, second element, and then I need to put my third element. Okay, this is another example. What the mistake on this table is the authors don't do the mean comparisons. So how to know that 2 and 4 are different or not? For example, in here you have ash 2%, protein 4%. Is it protein and ash is different? How to know? Because the author doesn't did the comparison, mean comparison. Okay, so suppose the author did ANOVA so that we can know Is it 0.8 and 2 were different or not? Is it 90 and 65 were different or not? So if like this, it's difficult for us to tell, right? So this is an example, another way to write the table. So they have the type of oil, and then they have the title on the top, okay, and then they say that means in brackets standard deviations, acrylamide concentrations in microgram per kilogram in deep fried sweet potato chips using different type of oil, right? So this is sample, and this is the mean. We know this is the mean. The bracket will be standard deviation, and the unit will be microgram per kilogram sample. And then at the top, they mentioned that A to C value within the same column with different letters are significantly different. So means that A is different, B is different. So canola and coconut not significantly different at the P less than 0.05. Okay. Palm oil and coconut is different. Palm oil and soybean is different. Palm oil and canola also different. Soybean and others are different because they have different numbers. So 1724, 1729 are not significantly different. So that's what we call it from the table. Okay, so how to write the result? This is an example. The table 3 shows. So this is the locations. The location of the result. There was a significant difference in acrylamide concentration between palm oil and soybean. So this is describe the result, right? Whereas in different in acrylamide concentration between coconut and canola was not significantly different. At P less equal to 0.07. So in this research they use P equal to 0.07, which is not significantly different. If the P less than 0.05 only considered to be a different. The mean acrylamide concentration was higher for soybean, and the lowest for only 1447. The mean acrylamide concentration for coconut was determined at this much and this much. So this is the detail up of the values. Still measuring your result, which is second element. So for the first discuss the highest, and second discuss the lowest. Okay. So how to write discussions? Either you arrange result discussion, result discussion, result discussion. If you have many results, I'm sure that you have many results in the thesis. It's not only one table or one figure or one results only. Should be have many. For example, if you do data collections, you can break down into many table figure. We don't put everything in one table or one figure, so that people confused to read. So if you do you break down into small pieces, but you have sequence that will make the story more clear. Okay, so write a thesis. It's like you tell people the story of your result. So how the story this should become one by one, kan? Tak boleh you nak langsam book everything to the readers, right? Should be one by one. For those who doing systematic analysis, also same. You have to do one by one. So this is your finding from the crude data. And then from the crude data, you do findings on the selected data after you remove all the, after you do exclusions, you remove all the not related journal and so on. And then from that one, only you find this is the main finding, should be in the table form. And then from that main finding, you find that this is the most important topic discussed by all the authors. Maybe if out from 20 journals, 10 topics, 10 journal discuss of the same topic. So that draw your attention to read in detail. Okay, so so 3 components should be there. And then how to write the discussion. Okay, either you combine result discussion, result comment, which is elementary, or you can tell all the result together. For example, this is all result together, and the last end should become with the comment. The last part is the comment. Okay, so how to write the elementary? We're going to continue after the prayer. Before we take a break, please in the table of figure presentation, you must have at least 3 elements to describe. The first element to describe the location of the figure or table. The second element to tell the result. Basically, we tell the one is you can see clearly from the result, or you can tell us data yang tersurat, not tersirat, tersurat. That you can see on the table. And then the element, you should discuss a bit why and how to explain a brief in the table, in the figure. Okay. The thing you must remember, talking about table and figure, the table and figure should be self-explanatory table or complete figure, right? What is the elements? First one, the title, the table on the top, the figure at the bottom. So that's the first one. The second element, the unit. Okay, either you put in percentage, you put in the value in the percentage, the value unit in the milligram per kilogram, sample, and so on. So the unit must be clear. The third element, it must have the statistical analysis. Either you do a t-test, either you do ANOVA, okay? And then what is the p-value for the statistical analysis? Okay, the fourth element should be any acronym. If you have any acronym, you must explain in detail at the bottom of the table or legends or bottom of the figure. If you have an acronym, so don't let the readers question or wondering what is the acronym for and so on. So just imagine the table and figure should be separated from the text. Means that if you take out the table or figure from the thesis and you read by its own, you can understand what is the message from the table or figure, right? So the table and figure should be self-explanatory. Should be complete. If you do a statistical analysis, should be have the alphabet, either is significant or not. Okay. If all the data is not significant, means that you don't have to put the alphabet. Just mention once, all the data was not significantly different. But if the data are different, for example, you have 5 group of sample, group 1 and group 2 different, group 3, 2, 3, 4 not different, 1 and 5 different. So means that you have to put different alphabet. And then there are also different way if you do one-way ANOVA or you do a two-way ANOVA, right? Basically, the alphabet will determine either you are comparing within the same column or within the same rows. So maybe you have treatment versus sample. So the treatment might be different, but the sample might be not different. So the alphabet will tell everything. Okay, should be clear on that one. Geraklah ni. Okay, how to write the elements? Either you write the result and comment, result and comment, result and comment, or you can combine all the result 1, result 2, and result 3, and comment at the last element after, not last element, element after result. Okay. Why is it? Why is the condition you can combine these 3 results together? Is it you can combine all elements, all the result, or you can you can separate all the result. The condition is, if you want to combine like this pattern, R1, R2, and R3 together, followed by comment, this R1, R2, and 3, they are the same thing, same result. For example, you talk about the proximity analysis. So R1 about the moisture content, R2 about the protein, R3 for the ash content. And you're going to have R4 for example, carbohydrate content and so on. So in that situation, you can combine the result. But if your pattern is different, for example, your result R1 talking about microbe, R2 talking about the physical properties of the food, R3 talking about the biochemistry property of the food, so you cannot combine, right? So it's become confusing. So it's better to split, into this element R1, C1, R2, C2, R3, C3. But if the thing talking about the same element, then you can combine. Another example of the same element. Okay. For example, R1 talking about the microbe E. coli in the running tap water. R2 result 2 talking about the E. coli in the well. R3 E. coli in the river. So all of them, they are talking about E. coli, but different area, right? So you can combine. Okay, so you have to know when to combine result and when to split the result. Okay, now we're going to write on the element 3, which is discussions of the result. So how to write discussion of the result of your findings? There are a few things you can do. This is just general rules. I don't say that you have to follow all, but just a guideline for you. If you don't have any idea how to discuss the result, this is what you can do. The first one, you can compare your result with others, right? So there's no, it's impossible you only the person who did the same research in that research in this world. For example, if I'm measuring the E. coli in water, so many researchers also reported the same thing. So I can use that one as my reference. Is it mine is lower or not, or higher or same value as reported earlier by others researcher, right? So the closest one is the best. What the closest mean? For example, I'm measuring the water in running tap water in Klang Valley area, right? So nobody report on the microbe content in the running tap water, in the tap water in the Klang Valley in this year, 2021. But I have one reference reported in the tap water, not in Lembah Kelang, but in Kelantan. So I can use that as my comparison. Or there is one paper reported the microbe content not in running tap water, but in the well or in the river in the Klang Valley. So I can compare my result with that that result as well. Okay, so the comparison should be follow the same result, and sometimes you can discuss in opposite. Macam mana nak discuss in opposite ni? For example, in your study, you got the microbe content. Bagi contoh microbe je. Sebab tadi ada bagi contoh microbe. The microbe content in the tap water is 100 CFU per ml. And then you found that another research found that the microbe content should be 1000 CFU per ml, which is 10 times higher than your findings. So it's two difference. It's not same, right? If other reported, you report 100, other report 110, 120, is still acceptable to be used as a comparison. But this guy reported 1,000 CFU per ml, which is 10 times higher than yours. So you can use or not as your comparison, because they are different. They are different too much. So the answer is yes, you can use, right, with possible explanation. Okay, for example, yours, you found 100 CFU E. coli in ml. That authors found that 1,000 CFU E. coli per ml, and then you must dig again. Dig mean that you have to know in detail why that happens. Maybe that guy that report 1,000 CFU per ml, they not, maybe the data was in 1983, which is at the time the chlorination, maybe the chlorination is not in place, right? Or maybe they take the sample from the well water, not the normal pipe. Okay, so must explain the possible reasons, not the same one. If you get the same trend, that's better. That makes your life easier. Okay, to explain the same reason. But if you have different reason, possible to be used as well as a comparison, but with the explanation why. Right? So when you already have the data, you already explain the data why, and finally what you have to do, you have to generalize your data to the populations. So you're finding just a bit, so you have to generalize the data, how it benefit to populations, right? So that should be your discussion. Okay, benefit the populations. For example, if you talk about microbe, is it the drinking water is safe or not to be used by population? So the example. Okay, all right. What is the element on the result? As I mentioned earlier, about 6 elements. One of them is statistical analysis. Either use t-test, either use ANOVA or not. And then people asking, how about those who are doing the survey and not survey? Survey also you have to do this t-test, correlation, regression, ANOVA. How about people are doing meta-analysis? What kind of statistical analysis they use? I'm not really expert in this meta-analysis, but I think there is a way how you're going to analyze the data. I think there has a protocol, so you have to follow that protocol, right? If you read in the many review paper, they will follow that protocol to analyze the data. Okay. The second element, how you're going to present the data, table or figure. So as I mentioned earlier, if you're explaining in detail about the values, so better to put in table form. But if you presenting about the trend, either the value increase or decrease, either you are comparing between A, B, and C, okay, or you are discussing about you are presenting about the effect of any treatments, and then you want to see what will happen if you change something. So better to put in figure form, right? So figure is better if you want to compare. And then don't forget to put the captions. All right, this is example of table. Correct. Example of table, the internet is unstable. Ini contoh dia. Right. So table 1, table 3, name. Then I have my my value there A. A is data, and I mentioned my replicates, it replicates times 3 sample, so means that 9. And then the second table, table 3, ascorbic acid content, a fresh and freeze dry sample. So star fruits. A, what is A? Data expressed in milligram per 100 gram of fresh week basis. Okay. And then values with the same capital with the same row are not significantly different between the fresh and freeze dried fruits. Data stand as mean of 3 replicates. So this is an example how you write the table, and then you will have this alphabet. So what you can tell from here, if this B capital, all of them are B capital, this is B capital as well. So means that there is no difference between fresh and freeze dry. But the small one, small capital, small letters. So D, this is C, this is B, E, E. So what you can tell, E, they are same. Muskmelon and watermelon, ascorbic acid content in fresh is same. This one also put same because they have E and E. B, C are different. B, E are different. B, D are different. C, D are different. C, B are different. C, E are different. Okay, so you can tell that muskmelon, watermelon, the ascorbic acid content is low as compared to the papaya. Papaya higher, mango, star fruit, watermelons, watermelon less. Star fruit, mango, papaya lah paling tinggi sekali, watermelons. Okay, this is an example. So don't forget to eat your papaya. Itu tinggi ascorbic acid content. Sama aja reading dia, boleh percaya tak? Then an example of the figure. You have the figure. So this is the title for the figure. Figure 2.1, gene expression analysis of several genes involved in evolution. Okay, the maturity stages of tomato include mature green, so that people understand what is MG, mature green. Yeah? On the top one, what is MG, mature green? What is TO? What is OR? What is red? So if you don't mention clearly at the bottom of this figure, what is OT, what is TO, what is OR, what is red, people are not going to understand. And then what is the data for relative gene expression? Okay, using housekeeping gene, the mean followed by 3 different letters, followed by different letter are significantly different. P point less than 0.05. Then this is PI3K. What PI3K, PI4K, PIP? What is this? So people don't know until they read these things. So this PI3K for phosphatidate and acetylcholine kinase, PI4K and PIP. Okay. So the table, the figure should be like this: self-explanatory, clear. People understand, and you can see that. Okay, A means that no significantly different, even though the value is lower. Okay, I think this comparison between sample between genes. Okay, A, even though this both are A, but the value is lower. Or relative between SKP. Betul lah. So how about this one? B. So this is A and B. This is A, B, and C. This is C, the higher. See? So A different between B, A different between C, and C different between B. So this is how we discuss the data. Right? Any questions? Ada soalan tak? Alright. So how to present the data? You must use the proper verbs. For example, if you are locating the table or figure, you have to use present tense. Okay, don't use past tense. Past tense means that you already complete the thing. So present tense means that you still ongoing reporting the data or table in that figure. All right. And then basically we put in the passive voice. It's very seldom we use active voice. It's possible, but it's depend lah. It's depend. You can use two mode, either in the passive verb, passive statement, or active statement. Right? Example: result of the t-test comparison are presented in table 1. So are presented dibentangkan. Ataupun active voice. Table 4 summarize the test of the result on the pre-contaminated insulators. So table 1 summarize. So more active voice. Okay, element 2 must use past tense. Always use past tense for the element 2 because that is your data, right? So your data means that you're finding, you found that you're not in the searching process anymore. You already found the data. That's why it should be in past tense. Example. Example, a group of divorced mothers spend over twice as much time in employment as married mothers. So spend past tense, right? Or you can say that was found. For the coefficient of correlation was found to be significant at the p, dah tinggal p ni, p less than 0.001 level. Okay, so was found, not is found, because this is your result. Okay, elementary. Elementary basically that will make your result more interesting to be read when you use proper elementary, which is a supporting data, right? Supporting data or reference or comparison. Remember, as I mentioned earlier, what is the characteristic of elementary? You can compare with others' data, right? Either you have the same trend, different trend, same value, different value. Never mind. Don't say that I have different value. It's not good. As long as you can explain why and why, it should be good, right? Even the geography factor also can influence the result. If you use different variety, if you use different technology, for example, A reported the the E. coli in running tap water, they use salation agar. Okay, in your study, you measuring the running tap water, you use PCR, right? The result might be different because PCR and salation agar, they are two different methods. Sensitivity are different. PCR is more sensitive than salation agar. Okay, so when write on the elementary, you can use past, not past tense, present tense. Past tense is only present tense or models, auxiliaries. Contoh model auxiliaries we're going to discuss after this. Okay, fine. Example. Okay, this is inconsistent with earlier findings. So is with earlier findings suggesting that personal characteristics are not related to the iterations and teaching. So is and are, they are the result from previous researcher, but you reported in present time. So because you bring the knowledge, the findings of the researcher is become, it's like a fact. So you are discussing the fact from the findings, so it should be in present time. Okay. Other method to be safe side, you can explain in auxiliaries model. When you explain in model auxiliaries, because you have to use can, may, could be, due to, may, because of that one, or you can say that could or should, right? So in that situation, you have to use the root word in the present tense. Okay. For example, the result can be explained. So can should be in present tense. It cannot be followed by. Okay. Right. And also you can use may. For example, the hyperactive children may be generally responsive to amphetamines. So modus auxiliaries is a safe way to discuss the result. Why you have to use modus auxiliaries? You don't use a fact, because the modus auxiliaries allow, if in future if somebody found what you say is wrong, it's not the real fact, you will be safe for a while. Why? Because you are using may. May means that could be possible. So you're not really sure. But if you use facts in explaining the things, if something happened to the fact, for example, using the theory and so on, the theory, and in future people found that the theory is not correct, something wrong. So that may affect your thesis. So if you don't use that, what we call that fact, you use model auxiliary, you can still cover the thing a bit, right? Okay, so let's look how to discuss in detail. Remember the topic of the result and discussion, okay, as compared to the literature review. The literature review begins with a big scale, and then you draw a bit to the funnel. Remember the funnel theory, right? You go funnel to funnel, and you get the methodology very detailed according to the funnel. And then now you're on the result and discussion. So on result and discussion is going to spread again. Dia macam funnel juga. Dia funnel up and funnel down. Gitu. Lansing, dia lansing. Alright. So in discussion, you're going to discuss your finding and try to relate to the population. Population means general people, general populations. Okay, so that should be discussion on your result. It should relate with the specific, you are doing a specific thing, relate with the general views of the readers. For example, if you talk about the E. coli in tap water, you can just go to the safety of E. coli, the safety of running tap water. So how it should save? Maybe you can mention also about the, apart from E. coli, there are also other hazardous in running tap water. For example, another microbe, which is you not mentioned in your study, that could be possible. And also you can mention also about the mechanism of E. coli, how E. coli going to affect when the consumer drink water contain a lot of E. coli and so on. But you don't do the data on that, right? There's no data. You don't collect the data, but you take from the literature. You correlate your findings to that situation. Okay, so this is how you can do it. Make it bigger scope at the bottom. Remember the funnel theory, literature review from the broad and specific to your subject matters, and then from that subject matter you're going to spread out again to the populations, and this is about it. You spreading the discussion part, and finally you lead to your general findings, which is can be used for all people, right? Okay, all right, how to write a discussions. Basically there will be 6 basic steps. Any questions, guys, before we continue? Ada soalan? Tak ada? Semua okay? Faham? Aina
Okay,
okay?
Doktor. Okay,
Yeah.
Doktor.
Right. Thank you. Okay. How to discuss? Discussion. Many students they are stuck in discussion. They don't know what to discuss. What should I discuss? My result, right? Why you don't know what to discuss? Because you don't know where is your result stand for. Okay, you don't know location. You don't know your hypothesis. You're not clear with your hypothesis. You don't know where is the research gap. If you have a very clear research gap, you know research gap is kind of puzzle, right? You just take one piece of the macam puzzle tu yang cabut-cabut tu, and then you know where to put it, you can do it very fast. But if you take one piece of the puzzle, you don't know which board you're going to fit in, and then you will take longer time to find what to describe yourself. So that's why it's important for you to think your title in the broadest piece, broader scope, not only specific to your title, but you should know in the bird eyes view what should be your title for, and then what is the gap. So when you know the gap, so you can make the thing easier to discuss, right? Okay, so how to do a discussion? The first step is you put a reference, or you mention again your purpose, your objective, your hypothesis. Remember what is your hypothesis again? You already mentioned in the objective, right? Before objective, you mention your hypothesis and so on. So you mention again here in this discussion part after you discuss your result. Okay, so mention again. Say that according to hypothesis, basically we don't mention the word hypothesis, but it is hypothesis. It can be suggested that the present the tap water in Klang Valley may not contain a microbe, may not contaminated with microbe because of the chlorination process. Okay, so that is your hypothesis. And then the second step is you review your important findings. Why is the important finding? So this has been supported in our finding, where we found that the number of E. coli colony in the selected tap water is below than 100 CFU, which is lower than permitted according to the Food Act 1983. Food Act 1983 suggested that the water should not contain E. coli more than 150 CFU per ml. Just example, don't take serious. Okay. So you explain again what is your finding, how your findings support the hypothesis. Either the important findings agree with your hypothesis or not. Okay. And then try to find other finding from previous researcher. Either they agree with your findings or they disagree with the findings. Okay. Step 3, try to explain possible explanation why. Macam saya kata tadi lah, I don't know in detail, but you should know in detail lah in your subject matter kan. Explain possible explanation, or you may speculate the thing. Is it okay or not to speculate? Some people don't like speculation much in the discussion before, because they say that you don't do that. How you sure? So that is the thing, right? But some people they allow to have speculation because they understand some of the limitations, some of the limitation of time and so on, make the researcher not able to prove this question is true or not. Okay, and then they allow for you. Okay, you can write, but you must use model auxiliaries. That's why I tell you it'd be safe for you to use model auxiliaries. Don't write as a fact, but write as a model auxiliaries, right? For example, may do itu could be explain do itu, so something like that. Okay, and then explain what is limitation of the study if you cannot link to that general findings, and then what is implication of the study to the populations. Remember, you have to broaden again the finding. So how the study, the data going to impact the population. For example, when you talk about the drinking tap water, if even though the number of E. coli low or lower than permitted in regulation, but there is still a potential risk. So it is suggested that all the people to boil drink the water before drink, or to use filtered water. Okay, using 0.2 micron filters for size, because of the microbe only cannot pass in through 0.2 micron of filter size. Or they can use other disinfecting techniques, for example, non-thermal technique, UV technique, and so on. Okay, so that will generalize your data. And then recommendation for future research. So in future, for example, in future, more research should be conducted on how the effectiveness of this disinfecting agent in activating the E. coli in drinking water. Dah siap, right? So you have complete one part of your data. Is it? Simple. 6 steps in data analysis. Any questions about the discussion part before we go to the conclusion part? Any questions, guys? Ada soalan? Fazil, ada soalan?
Ada doktor.
Tak ada. Okay. So if you don't have any questions, so means that, okay, when you present your data, you have a data table or figure, discussion, step 1 to 6, and conclusion, right? And then table 2 or figure 2, discussion 1 to 6, and conclusion. And then you're going to have table 3 or figure 3, discussion 1 to 6, and conclusion, right? So that conclusion is for topic conclusions. But what is conclusion for your thesis? Okay, so the conclusion of the thesis basically should answer your hypothesis. They should answer your hypothesis, right? Okay, so how to write conclusion? This is the final step of your thesis. So you should answer your hypothesis. Either the hypothesis is correct or not, either your objective is achieved or not, either your findings answering your objective. Okay, so that should be in conclusions, right? So how you conclude, it should be backed up by your data. So data should be back up your conclusions. And then basically in the first part of the conclusion, we're going to tell again what is our hypothesis, what is our problem statement, and then followed by your findings, your findings, the answering the hypothesis, the answering the problem statement. Okay, what are the mistakes during conclusion? Some people, they like to summarize all the data. Summarize will be different with conclusion. They summarize all the point in the data, and then they list again in this conclusion. For example, they say that table A, chapter A found this one, chapter 1 found this one, chapter 2 found this one, chapter 3 we found this one, chapter 4 we found this one. So you can use that. That's point, because conclusion is not the way, the place that you summarize. You repeat again the same statement that you write in the result and discussion. And then when you write the thesis, try not to use the word we, or they call as suku kata kedua. You must use, is it suggested for you to use the word third party? For example, the research found that, the study found that. It's not you or them, okay, but it's about third party, macam tu. Right? So don't use we found that. It's not really good way to write the thesis. So better to just mention the authors found that. Okay, not they found that, the authors. And then the study found that, not for example, I'm citing Nurfazi. Nurfazi did a research. Nurfazi et al. 2020, they found that. So not really nice way to write the thesis. You should write Nurfazi et al. Found that. Okay, the authors describe something like that. Right? So this is how to write conclusion. Some of the conclusion, they are too detailed. Tadi they write every single findings in each chapter, but some of them they report too detailed. For example, you're reporting about proximate analysis. So you come again in the conclusion, you write again the values. By right, you already write in the literature, in the result discussion. So don't write again the same numbers in the conclusion, right? So just summary your findings, okay? And then sometimes you're going to introduce a new idea in the conclusions. So what does it mean? Means that your study is not complete. There's a place for you to introduce a new idea. They call as future recommendation, but not in the conclusion. The future recommendation is considered to be the next step. What people have to do from your data, your suggestion, you suggest people to do this experiment and so on. So that will be on future recommendation, not in the conclusion, right? So don't put in conclusion. Right. SEL lagi. Okay. Anybody have your own your conclusion now? Ada tak conclusion? SEL je kan Dr. Sabri ni. Tak ada kerja lain. SEL. Any question before you go to the SEL? Last SEL.